Good evening, and I welcome you all to the week 5 live session of this NPTEL course, Experimental Biochemistry. I am Surasree. I am a Prime Minister's Research Fellow working in the Department of Biotechnology, IIT Kharagpur. Let's begin today's session. In today's session, we will discuss about several protein purification techniques or several chromatographic techniques. These will involve size exclusion chromatography, ion exchange chromatography, affinity, hydrophobic interaction chromatography, and dialysis. So, we are going to talk about protein purification. Now, we need to understand why do we need purification of proteins. So, suppose we want to work with a protein and we want to know its structure, its function, its characteristics. So if we have to do that, so what we usually do is we overexpress this protein in a particular cell system. It can be bacterial cell, it can be yeast cell, it can be mammalian cells as well. So what we do is we overexpress the protein in that particular cell type, and then we purify this protein from all other cellular proteins. So that particular organism, either it can be E. coli or it can be yeast, will also express its own endogenous proteins along with your protein of interest. But what you need to do is you need to separate your protein of interest from the rest of the proteins. So how do we do that? For that we apply several chromatographic techniques. But the basic of protein purification begins with tissue disruption. So the cell in which you are expressing your protein, that tissue, first you need to disrupt. So what you do is you dissolve that tissue, dissolve the cell membrane to allow all the components of the cell to come out in the solution. So that is how we destroy the cellular architecture and all the components of the cell come out in the solution. But this solution will contain proteins as well as all other cellular debris. So how do we then separate the proteins from them? So for that we do crude fractionation. So for crude fractionation we often use centrifugation. So this is a technique we will discuss later on in today's session. By centrifugation you can separate the proteins from the cellular debris. Now followed by that we apply selected fractionation. So this selected fractionation is selective based on my protein of choice and based on the technique by which we want to purify our protein. So these actually involve chromatographic techniques, and based on the characteristic of the protein by which we want to separate it or purify it from other proteins can be actually size or mass of the protein, for which we apply gel filtration chromatography. It can be charge of the protein, for which we usually use ion exchange chromatography. It can be hydrophobicity of the protein. So some proteins are more hydrophobic than others. So depending on that, we use hydrophobic interaction chromatography or reverse phase chromatography, and also binding affinity of the protein. So it is actually affinity chromatography. So what we do is we take help of the binding affinity of the protein to a specific biological molecule. Now these are several techniques which we are going to discuss in today's session. Now let's begin with centrifugation. So this is a crude fractionation technique. So what we do in centrifugation is that we actually take our tissue homogenate. So we dislodge the tissue, we actually dissolve the tissue, and then we rupture the cell membrane in particular ways. So first thing that we can do is we can sonicate the sample. So sonication is using ultrasound to break down the cellular architecture, the cell membrane. Then after sonication, what happens? The components of the cell come out in the solution, so that is the tissue homogenate. So now what we do is we can centrifuge the whole sample. We can centrifuge it in several speed, and depending on the speed of the centrifugation, the heavier components will actually come out in the pellet, so they will precipitate, and the lighter components will remain in the solution, and that will depend on how much speed we are applying. So if we centrifuge in low speed, what we will see is that the pellet will contain whole cells, the cell debris, the nuclei, cell cytoskeletons. Then again, if we centrifuge at a medium speed, then the pellet will contain these organelles, mitochondria, lysosomes, peroxisomes, etc. Then if the supernatant of this particular centrifugation is again subjected to very high speed centrifugation, then the very small organelles like microsomes, the small vesicles, they will also come in the pellet, right? And then followed by very high speed centrifugation, which is also known as ultra centrifugation. What we will get is that in the pellet we will get ribosomes, viruses, and large macromolecules. So this is differential centrifugation. So you actually vary the speed of centrifugation to separate particular elements of the cell in pellet from the rest of the elements, right? So this is how you centrifuge. So for centrifugation we use a machine which is called a centrifuge, and this centrifuge machine can have different types of rotors. So actually there can be fixed angle rotors, there can be swing rotors. So what happens in fixed angle rotor and what is the difference in swing rotor? The fixed angle rotors rotate at a particular angle, that is why it's called fixed angle. And in swing rotors, the tubes actually become horizontal. So these are the tubes in which we are putting the sample, and the tubes become horizontal in swing rotors, and that is how the centrifugation is carried out. So suppose we are expressing our cells in Escherichia coli or E. coli bacteria. We are expressing our protein in E. coli bacteria. So what we do is, after we overexpress the protein, we collect the cells, and then what we do is we pellet down the cells, or we centrifuge the cells first. So then what happens, the whole cells, it comes in the precipitate. So you can see for that we need medium speed centrifugation. So it can be around 6000 rpm. rpm is rotation per minute. So if we centrifuge the cells at 6000 rpm, then the cells will come in the pellet, okay, and the media we can discard. So the cells, that is how we can pellet. Now what we do next is, so this is pelleting the pellet bacterial cells. Next thing what we can do is we can dislodge the pellet or dissolve the pellet. Next, after dissolving the pellet, we again allow it for centrifugation at 11,000 rpm. So this is high speed centrifugation. So at 11,000 rpm, then what will happen? The proteins will come in the solution and the cell debris will be in the pellet. Cell debris will be in the pellet, right? So this is the first step of protein purification. Now when we get these proteins in solution, now we need to subject this solution to chromatography to separate our protein of interest from rest of the proteins. So next step is chromatography. So this is physical separation based on the differential migration of analytes in a mobile phase as they move along a stationary phase. So this is the separation between a mobile phase and a stationary phase. So what is a mobile phase? A mobile phase is the phase which is moving, which is mobile, and the stationary phase is usually the matrix on which we are performing this chromatography. Now there can be several mechanisms of separation. So it can be partitioning, it can be absorption, exclusion, affinity, or ion exchange. We will discuss about these modes of separation. But first let's talk about the partition coefficient. So the partition coefficient is the distribution or partitioning of solutes between mobile and stationary phase. So it can be represented by K equals to Cs by Cm. Cs is the concentration of the solute in the the concentration of the solute in the stationary phase divided by the concentration of the solute in the mobile phase, right? So that determines how much solute will actually be coming in the mobile phase and how much solute will remain in the stationary phase. So that actually determines our efficiency of purification or chromatographic separation, right? There can be another method which is salt fractionation. So what happens in salt fractionation is that at low ionic strength of salt, increasing salt concentration tend to increase the solubility of the protein. So this is called salting in. Salting in is the event when when you increase the salt concentration, then what happens? It increases the solubility of the protein. Salt concentration increases, solubility of the protein also increases. Another event is called salting out, which is, so this occurs for a certain till a certain concentration of the salt, but after that, after this point, then what happens? As you increase the salt concentration, the solubility decreases. So salt concentration you increase, but the solubility of the protein will decrease. So that is called salting out of the protein. So the protein will actually be denatured in this kind of condition. Okay, so that is salting in and salting out of the proteins. So next we will look at a particular type of chromatography, which is gel filtration chromatography, or also called size exclusion chromatography, or SEC. So this chromatographic technique involves the separation of the proteins based on size and shape of the protein. So the protein size is actually the molecular weight of the protein, and molecular weight of protein is measured in daltons or kilodaltons. So what is the principle of this gel filtration chromatography? That is, so we have a protein sample where we have proteins of multiple molecular weights. Okay, so there can be proteins of, for example, 20 kilodalton, there can be proteins of 80 kilodalton, and there can be also proteins of 200 kilodalton. So I'm writing here, that is protein A, protein B, protein C, and protein D. So we have, suppose, proteins of these many molecular weights, these type of molecular weights mixed in a solution. So how do we separate these proteins based on its size or its molecular weight? So what we do is we allow the proteins to run through a gel filtration column. So this is a column, this is actually a gel filtration column, and what you can see here is that there are porous gel beads inside the column, packed inside the column. So now what we do is we first pour the sample. We actually load the sample in this particular column, and this column will have small molecules as well as large molecules, right? So large and small based on molecular weight. Now what will happen, these porous gel beads, they have particular size. So they have particular pore sizes which actually has a cutoff. So based, what is the importance of this cutoff is that these pore sizes will determine which range of proteins can be resolved in this particular gel filtration column. Now now these proteins, they will pass through this column, and in these porous gel beads, actually the proteins can remain, or protein can go inside these porous beads, in the pores of these beads. So therefore what will happen, the small proteins, they can easily go inside the beads, right? They can easily go inside the pores of these beads, right? But the large protein molecules, they will not be able to get inside the beads of the get inside the pores of the beads, right? So this actually will depend on what is the cutoff, molecular weight cutoff. So above that, the proteins we will not be able to resolve. So what will happen, these large molecules will come out first. So the protein which has the highest molecular weight, this protein A will come out first. So this is a chromatogram which we can observe, where this x-axis is the retention time, and the y-axis can be parameter which can be UV absorbance, which can be conductivity, which can be pH, etc. So in this retention time means what? So basically at what time which some molecule is being eluted, that is the retention time. So how much time it is retained inside the column. So large molecules will have smaller retention time, so they will come out earlier, like A and B. But then what will happen, as we pass more and more solvent or more and more buffer through it, then the small molecules will also start to come out. So C, which has a 90 kilodalton weight, will come out first, and followed by D, which has a lesser molecular weight than C, right? So this is how, because smaller molecules will have higher retention time, so they will remain more time in the column and come out or elute later. So this is the principle of gel filtration chromatography. Now this is how the system actually looks like of a gel exclusion or gel filtration chromatography or size exclusion chromatography. So you have a solvent reservoir. So where you are providing either water or buffer or ethanol. So why ethanol? Because for cleaning the column and storing the column, we usually store the column in 20% ethanol. But for first, what we do, if we have to purify using a gel filtration chromatography, we run water in the column. So we first wash the column with water. So in that time, in the solvent reservoir, we will keep water and we will run water through the column. Now next, what we will do is we will equilibrate the column with the elution buffer. So the buffer in which your protein will be eluted, in that buffer we will equilibrate the column. That is a very necessary step for purification, before purification actually. So in this time we will keep the buffer in this solvent reservoir. Next, what we will do is we will use a pump. So why is a pump used? Because in this column we need to maintain the rate of flow of the solvent through this column. So pump will maintain the rate of flow or the flow rate. Now the flow rate has to be monitored very carefully, because if the flow rate increases, then it will create a back pressure in the column, because the column is tightly packed. So there will be a back pressure in the column and the column will get damaged because of the high flow rate. So we need to keep the flow rate in check. Now the next thing is, this pump will allow a particular flow rate of solvent, and that will pass through the column. So now there is an injection valve also, so where you can inject your sample. So you can inject your protein sample in this injection valve, and that will be taken to the column, and then whatever comes out from the column goes through a detector. So this detector is usually an UV absorbance. It detects UV absorbance. So by which we can monitor that what is the like how much protein is being eluted at what time, right? And this detector will send the signal to a monitor or recorder where we will be able to observe the chromatogram. So where we will actually get the chromatogram, right? So from the detector it will go to the monitor, and then the protein or the solvent will actually it can be either collected. So we have to set the system, the FPLC system accordingly, where we can collect the protein or we can collect it in waste. So that means something which is not our required protein, we can collect it in waste. So this is the FPLC purification system. This is the general schematic of this system. Now let's talk about the partition coefficient of the size exclusion column. So this is how a typical size exclusion column looks like, and there are certain terminologies associated with it. One is a void volume and total volume. So total volume, you can understand, total volume is the total volume of VT is the column volume, the total volume of the column. Whereas what is V0 or void volume? Void volume is the volume which is unoccupied by the porous beads, right? So you can see that there is a portion, there is a space between the porous beads, right? So this is the volume which is unoccupied by the matrix or by the beads, right? So that is actually your void volume, and then the final or the full volume of the column is the total volume. So void volume, there is a rule which states that void volume is actually 1/3 of total volume or VT, right? So void volume is 1/3 of total volume. So suppose your column volume is 120 ml. So what will be your void volume? Your void volume will be approximately 40 ml. 1/3 of your total volume is your void volume. And Vs is the volume of the stationary phase. So stationary phase means the volume which is the volume of the porous beads, right? Now so now this partition coefficient, or the partition between the mobile phase and the stationary phase, is given by this particular equation, which states that K average, or the partition coefficient, is equal to VE minus V0 divided by VT minus V0. So it is VE. What is VE? VE is the elution volume of your analyte. So suppose your protein is being in a column volume of 120 ml. Suppose your protein is being eluted at 80 ml. Your void volume is 40 ml. Your total volume is 120 ml. So from this information we can calculate the partition coefficient. That will be VE minus V0, which is 80 minus 40, divided by VT minus V0, 120 minus 40. So this will give us the partition coefficient. That means how much protein is going to be partitioned in the mobile phase and the stationary phase, right? So this is the calculation. Now the particle size, the size of the beads, that will determine V0, that is the void volume, and the pore size of these beads will determine the dissolving range. So there are various resins. For example, there can be Sapphirose resin, there can be Sapphirose 200, Sapphirose 75. What are these 200? What are these 75? So this is the molecular weight cutoff. The 200 means 200 kilodalton. So the Sapphirose 200 resin will be able to, or the Sapphirose 200 beads, they will be able to resolve the proteins which are less than 200 kilodalton. Sapphirose 75 similarly. There can be other types of resins also where there are definite pore sizes. So this pore size is controlled, monitored, and that will actually determine the range, the resolving range that we can get in this particular column. And the K average, that is the partition coefficient, this will be proportional to the molecular weight of the protein in the resolving range. So the partition coefficient is proportional to molecular weight of the protein in the resolving range, remember. So actually in this range where the column can resolve proteins between two proteins, in that range this partition coefficient will be proportional to the molecular weight of the protein. Now why is this information necessary? Because in while we are running the size exclusion chromatography, we can also determine the molecular weight of unknown proteins by using size exclusion chromatography. How do we do that? So actually there is a standard curve of every column, of every size exclusion column. And this, how is this standard curve or calibration curve done? So actually we take several proteins like this, which are known proteins, and the molecular weight of these proteins are known. right? So for example, the proteins are aproteinin, um, RNase A, carbonic anhydrase, ovalbumin, conalbumin, aldolase, ferritin. These are the proteins of which we for which we know the molecular weight. Now what we do is we run the proteins in this column. We mix the proteins and then run this protein through the column. So first of all, what we do is we use blue dextran to determine the void volume. So because you can understand that what will come in the void volume. In this void volume, the molecules which are eluted, suppose this is the void volume of the protein. So if this is total 120 minutes or total 120 ml of solvent that is passed, so the first 40 ml will be void volume, which means the proteins which are not being able to come into the beads, come inside the beads of the pores of these beads, they will be eluted in this void volume. So the proteins which cannot go into the pores, that will come in the void volume. Anything that cannot go into the pores will come into the void volume. So therefore this void volume actually can determine by passing blue dextran through the column. So the blue dextran will come in the void volume. Now there are these proteins which are also passed through the column and their molecular weight is known. So now what we do is we have a chromatogram where the volume of the solvent is given with the with these peaks that you can see, right, with respect to the volume of the solvent. Now these peaks are the peaks of the absorbance of particular protein molecules. Now we know we can plot it that which protein is coming out at which volume, because these are known proteins, these molecular weights of these proteins are known. So we know which proteins are coming out in which volume, and what we will do is that we can plot it. Or so actually what we have here is that we know the elution volume of these proteins, right? So after running it in a size exclusion column, we get the information of the elution volume, that is VE, of these proteins, right? Now from the elution volume, what we can calculate? We can calculate the partition coefficient, which is VE minus V0 by VT minus V0, right? So V0 and VT, these are constant for a particular column. Now we have, if we get to know the VE, then we can calculate the partition coefficient. And now what we can do is we can plot this partition coefficient with the log of molecular weight of the proteins. Okay, the partition coefficient with the log of molecular weight of the proteins, and this will give us this kind of a graph. Now this is actually the standard curve of this graph, the calibration curve or the standard curve of this graph. Now if we are now purifying an unknown protein in this size exclusion column, then what happens? So when the protein comes out, we note down the elution volume. So we track the elution volume, and then what we do is we can plot it in this standard curve. We can plot the elution volume of the unknown protein in this standard curve, and by that we can determine the log of molecular weight. That means from which we can get an idea of the molecular weight of the protein. So from the elution, so suppose our elution volume is 80 ml. So from that we can calculate the partition coefficient. Now from the partition coefficient, suppose the partition coefficient comes as 0.4. So we plot it on the standard curve. And from which, if we extend this point to the x-axis, we will have an idea of the molecular weight of the protein. Right now, your protein of interest, the one which you are purifying, it can exist in several oligomeric forms. So it can exist as a monomer. There are proteins which can exist as dimers or tetramers, or several other types of oligomers as well. So in that case, the protein which will be coming out from the size exclusion column, if it is a dimer or if it is a tetramer, then its molecular weight is increased, right? So if it is, suppose we have a protein which has a molecular weight of 40 kilodalton. If the protein is a dimer, then we will have a molecular weight of 80 kilodalton. If the protein is a trimer, we will have a molecular weight of 120 kilodalton, right? So this is a monomer, dimer and trimer, or there can be several other oligomeric forms as well. Now from this standard curve, we can get an idea of this molecular weight, right? And suppose we have a theoretical value of the molecular weight of our protein, and that is 40 kilodalton. And now if we get the elution at a higher volume, which is for example, which is corresponding to the molecular weight of 80 kilodalton, then we can predict that yes, the protein is existing as a dimer in the solution. So it is being purified as a dimer. So if the protein is a monomer, that is a protein its molecular weight is 40 kilodalton, so it's a smaller protein, it will be eluting later, right? So its elution volume will be higher. But if the protein is eluted at a lesser volume, that means it is eluting out faster, right? Then what will happen? Then of course this protein has a higher molecular weight, and this higher molecular weight can exist from oligomers, or it can be the reason of or the oligomeric formation can be the reason of this earlier elution of this protein, right? So from which we can actually determine this oligomeric condition of this size of this protein, and also the molecular weight of this protein by size exclusion chromatography. So that's all about size exclusion chromatography. Next we will look at ion exchange chromatography. Ion exchange chromatography is a separation, chromatographic separation, which is based on the charge of the protein. Now ion exchange chromatography is very simple. It has a matrix which is either positively charged or negatively charged. So ion exchange chromatography is of two types: anion exchange or cation exchange. Okay, ion exchange chromatography can be of two types: anion exchange or cation exchange, which means that the matrix can be either positively charged or negatively charged. So if the matrix is negatively charged, that means the beads are negatively charged, then the positively charged proteins will bind to the negatively charged beads by electrostatic interaction, right? So the negatively charged bead will bind to the positively charged proteins, and the negatively charged proteins will not be able to bind in this matrix because of repulsion, charge repulsion. Therefore these these negatively charged proteins will come out in the flow through. So this is what happens when there is a negatively charged bead. Okay, if there is a negatively charged bead, which means that it is a cation exchanger. So the matrix is anionic, so which means it is a cation exchanger. So here we can see the example of these several beads or several matrix which are used in ion exchange chromatography. So these two, the first two, you can see that this one is a sulfate based resin. Here you can see this sulfur molecule. This is a sulfate derived resin, and here you can see that there is a negative charge, right? Sulfate is negatively charged, so you can see there is that is the matrix will be negatively charged. This is usually agarose. These are agarose beads. Okay, so the matrix, you can see that it is linked, the agarose bead is linked to this negatively charged group, because of which this matrix will be negatively charged. Similar with this, in this particular resin also, you can see that it is a carboxymethyl resin. So CM or carboxymethyl resin, there also you can see that since because of this presence of carboxy group, it is also a negatively charged resin. So now if this is since these two are negatively charged resin, so positively charged protein will bind to it. So positively charged protein means cationic proteins will bind to it. So these are cation exchangers. These are cation exchangers. So which means that the positively charged proteins will bind to it. Now how do we elute these proteins? We will use a cation of higher ionic strength to pass through the column, which will elute the proteins. So a stronger ionic strength cation will actually displace the protein molecules, so by which we will elute out the proteins from the resin or from the matrix, from the column. So these are cation exchangers, and the next two, these are anion exchangers, and which is why the matrix is positively charged. So you can see there is positive charge on the matrix. So this is a Q resin. It's usually called Q saccharose, and this one is a DEAE cellulose or DEAE saccharose. DEAE is diethyl aminoethyl. So here also you can see that there is a positive charge on this matrix. So this is a strong anion exchanger and this is a weak anion exchanger. So what will happen, that anionic proteins will bind to this resin, and the positively charged proteins, the cationic proteins, will come out in the flow through. Now for elution of this anion, again we need to pass a buffer which has a higher ionic strength of salt, which will displace the protein. So higher ionic strength cation or higher ionic strength anion will displace the protein molecules, and the protein molecules will be eluted out from the column. So this is cation and anion exchange, and this binding affinity of the proteins to any anion exchange resin will actually depend on several factors, like the concentration of the salt ions, and so the buffer in which we are purifying the protein. Suppose we are purifying the protein in tris HCl buffer, which is of pH 7, and we are using 1 molar NaCl for elution. Okay, so what will happen, that in this particular buffer concentration, in this particular buffer, what is the concentration of salt, that will actually determine how much will be the binding affinity of our protein of interest to this ion exchange resin, and also on the pH of the mobile phase. So how is this pH important here? So since the proteins are now charged, either they are positively charged or they are negatively charged, so therefore their pH or the pi of these proteins now also come under consideration. So let's see how. So suppose we have two proteins, A and B. Protein A has a pi of 4 and protein B has a pi of 9. Can we resolve these two proteins by ion exchange resin? Yes, we can. So suppose the pH, so therefore we need to select the pH of the buffer very carefully in which we will carry out this elution. So let's see how. So suppose we need our buffer contains a pH of, buffer pH is 7.4, right? If buffer pH is 7.4, then what will be the charge on protein A and what will be the charge on protein B? So if we know that if pH is less than pi, then protein is positively charged or protein exists as a cation, and if pH is greater than pi, then protein will exist as an anion, right? So in this pH 7.4, at pH 7.4, protein A will exist as a cation because it's at pH 7.4, which is pH which is less than, the pH is greater than the pi of the protein. So pi of protein A is 4, so pH is greater than pi. So in this pH, therefore the protein A will exist as an anion, and at pH 7.4, protein B has a pi of 9. So here pH is less than pi. Here pH is less than pi. So therefore in this pH, the protein B will remain positively charged. So therefore the protein A is an anion and protein B is a cation, right? Now we will have to decide which matrix we use in this case. So suppose we are using a Q-Sephiroth matrix. I told you in this slide that Q-Sephiroth, here Q-Sephiroth is actually, Q stands for quaternary. So Q is actually positively charged. So this matrix is positively charged. So Q-Sephiroth is positive. So therefore the anionic protein or protein A will bind to this Q-Sephiroth. Protein A will bind to the Q-Sephiroth and protein B will come in the flow through. Protein B has a positive charge, the matrix is also positively charged, so protein B will come in the flow through. Now we have A which is bound to the positively charged matrix. Now if we use another matrix which is, suppose carboxymethyl cellulose, CM cellulose. CM cellulose is a negatively charged matrix. So in this negatively charged matrix, the positively charged protein will bind, which is protein B will bind, and protein A will come in the flow through. Right? So now how do we elute these proteins out? The elution will depend on the buffer of higher salt concentration, or if we change the pH of the buffer. By that way also we can elute out the bound protein from the column. And we can separate these two proteins, protein A and B, based on their pi and based on their charge. Right? But remember, this is in this ion exchange resin. If the proteins are they have very similar pi, then these proteins will not be able, it is not possible to resolve them using ion exchange. So if the proteins have very similar pi, then we have to use other chromatographic methods. For example, we can use gel filtration chromatography if the protein is the proteins have different molecular weights. So if one protein has a very high molecular weight, another has a small molecular weight, then these two proteins can be separated by gel filtration chromatography, but not by ion exchange chromatography if they have very similar pi. Similarly, in gel filtration chromatography also, if the proteins have very close molecular weight, then they cannot be separated using a gel filtration column. So this is the ion exchange chromatography. Next we will talk about affinity chromatography. So this affinity chromatography is a very specific, it depends on a very specific biological interaction between two molecules. So what are these molecules? So there is actually a matrix, an inert matrix. So this is an inert matrix, and we have ligands associated or attached to this inert matrix. These ligands can be molecules which have specific interaction with a molecule which we want to isolate or which we want to separate or purify. So suppose if we attach insulin protein as a ligand on the agarose beads, then and we have a protein sample where we have the insulin receptors along with other proteins. So now if we run this protein sample through these agarose beads, then what will happen? This insulin receptor will actually attach to the insulin molecule by highly specific biological interactions, and therefore the rest of the proteins will come in the flow through, but only the insulin receptor will go and attach to the ligand, right? So this ligand and matrix has to be very carefully selected for this successful execution of affinity chromatography. So here we can see that if there are different types of proteins and we have a particular ligand associated with these beads, right? These are the ligands, right? Now this has a affinity, this is the affinity resin with the ligands attached. And now what we do is there is a target protein and there are other proteins which you do not want to separate, but we want to purify our target protein from it. So our target protein actually binds to this ligand, and it has a specific affinity for this ligand, which is a high affinity. Okay, so now we pass this protein sample through this column. Now therefore what will happen? Our target protein will bind to the ligand, and then what we do, we wash the column. We wash the column so that the proteins which are not bound to the matrix can come out in the flow through. So these proteins are coming out in the flow through which do not have any specific interaction with this ligand, and then we elute the target protein, which actually we do by competition. So what we do is we can pass a compound which competes with our target protein to interact with these ligands. Or what we can also do is we can change the pH of the solution, pH of the buffer, so that the target proteins cannot anymore bind to the ligand, and then it will come out in the solution, or it can come out in so it will be eluted out. So by which, these are the methods by which we can elute our protein. So this actually relies on this affinity between the ligand and the target protein, which is why it is called an affinity chromatography, and this interaction has to be reversible. So it should not be an irreversible interaction. If that is irreversible, then we will not be able to elute our protein of interest after it binds. So that means these interactions has to be, that is why they have to be reversible interactions. Now we use several activated gel matrices or the activated agarose. For example, cyanogen bromide activated agarose, where the primary amino groups can attach to it. Carbonyl diimidazole activated agarose or supports, which can actually where the N-alkyl carbamate groups can attach. Epoxy activated agarose, where hydroxythiol or amino groups can attach. So these agarose beads, they are designed such that there can be specific molecules which can attach or bind to these ligands by and allow to resolve the target protein or purify the target protein from the pool of proteins. Now, a very commonly used method is immobilized metal affinity chromatography, which is called IMAC. Immobilized metal affinity chromatography. So here what we do is we use nickel ion. So this metal affinity is, the metal here is nickel, or it can be cobalt. So what is this resin? So it is called a nickel NTA resin. Nickel NTA actually NTA stands for, so this is actually NTA. It is nitrilo triacetic acid. This is actually NTA, nitrilo triacetic acid. And so the importance of this kind of a resin is that it has a metal ion associated with it, which is nickel or which is cobalt, which can form coordination compound, which can form coordination complex. So how does that form? So actually what we do for this type of immobilized metal affinity chromatography, we express our proteins with 6x histidine tag. 6x histidine tag. So what is that? So this is a tag which expresses 6 consecutive histidine residues on the N-terminal or the C-terminal of the protein. So there are specific vectors where there is this tag incorporated that expresses 6 consecutive histidine residues in frame with the protein, so that the protein can have this 6x histidine tag at its N-terminal or at its C-terminal. Now what is the importance of the 6x histidine tag is that these histidine molecules, they can, I'm sorry, this is not the NTA, this is actually the NTA. So this is actually the NTA structure, which is nitrilotriacetic acid. So which is attached to this matrix, an inert matrix, right? So this is nitrilotriacetic acid, which has this nickel metal. Now our proteins which have this 6x histidine tag, they these histidine amino acids, they have this imidazole ring, right? They have this imidazole ring in this structure, the histidine amino acid structure. If you remember, they have this imidazole ring. Now what happens, these imidazole rings can form coordinate bonds with this nickel metal. You can see that these histidine molecules can form coordinate bonds with this nickel atom, and in this nickel metal, actually you can see that there can be 6 coordination bonds that can form with this nickel atom, and its 4 bonds are actually occupied by this matrix. 4 bonds are occupied by this matrix itself, but 2 bonds are open. So these two bonds are actually formed with the histidine residues of the protein. So since our protein contains 6 consecutive histidine residues, so these histidine residues can bind to this nickel atom very specifically with the formation of this coordination complex, and by which our 6x histidine tagged protein can bind to this matrix. So this is the metal affinity, the affinity for this nickel metal, or here there is cobalt metal also. So this is cobalt carboxymethyl aspartate. So this is cobalt carboxymethyl aspartate, this is the resin, right? And the histidine tagged, again the histidine tagged protein, these histidine residues, these are the histidine residues, right? These histidine residues, they have this imidazole group which can form coordination bond with this carboxymethyl aspartate resin, or here the NTA resin, and it can specifically bind to this resin with high affinity. Now the next question is, then how do we elute out this protein from the resin after it binds to the resin? So for this what we do is, first what we do, we take the column which is a nickel NTA which we filled with a nickel NTA resin. Now we wash the column with water, then we also wash the column with a binding buffer. So binding buffer is done for equilibration. So we equilibrate the column in which we have our protein sample. Now we next load the protein sample and we collect the flow through. So all the proteins which cannot bind to this resin will come out in the flow through. Now the proteins which are already bound, it now remains inside the column. So now what we do is we actually wash the column with sequentially higher concentration of imidazole. Elute with imidazole. So imidazole is a compound which will actually compete with these proteins. So imidazole actually competes with these proteins. So that is why what we do, because you can see that this protein also have this imidazole ring, right? And if we use increasing concentration of imidazole, and if we wash the column with increasing concentration of imidazole, so this imidazole will compete with the protein and it will allow the protein to detach from the matrix. So what we do is, for example, we can increase the concentration of imidazole with from 30 millimolar imidazole, 50 millimolar imidazole, 100 millimolar imidazole. So we make separate buffers of these imidazole concentrations, and we will pass this buffer of increasing imidazole concentration through this column, and at a particular imidazole concentration our protein of interest will be eluted from the column, and that we can finally run in the SDS page to check which concentration or in which concentration of the imidazole our protein is being eluted out. So this imidazole actually competes with the protein and allows it to detach from the matrix. So here we can see that we have several proteins, and the proteins which have this hexahistidine tag H6, they will only bind to this nickel matrix, and then we will have to elute this protein out, and if we elute this protein out with imidazole, then we will have these purified complexes. We will have these purified complexes. Now that is affinity chromatography. So next is hydrophobic interaction chromatography. Okay, one thing that I forgot to tell you, in affinity chromatography there can be several other types of affinity chromatography as well. For example, if we want to purify an antibody, so what we do is we attach them, or we link the antigen with the agarose for purification of the antibody. So our antibody, which will be specific to this antigen, because this specificity comes from the antigen antibody interaction. So this antibody, if it is specific to the antigen, it will bind to this antigen, and then we will be able to purify it by changing the pH of the buffer. And then if it is an enzyme, for example, if you want to purify an enzyme, then we can attach the substrate of the enzyme, we can attach the inhibitor of the enzyme to the agarose, so that we can purify the enzyme, specific enzyme, from the pool of proteins. So actually so you can see that in these types of purification, in affinity purification, it depends on the highly specific or high affinity interaction between the enzyme and the substrate, or the enzyme and the inhibitor, like antigen antibody interaction. So these specific interactions are utilized in this affinity chromatography, whereas we had discussed that in ion exchange chromatography, the ionic charge of the proteins are utilized for separation. So either it is a positively charged protein or it is a negatively charged protein. Depending on that, we use either a cation exchanger or anion exchanger. And then there was this particular protein which was this particular technique where in gel filtration chromatography, so where we utilize the size or the molecular weight of the protein for purification of the protein. Now this is another type of chromatography where the stationary phase is non-polar. The stationary phase is non-polar or hydrophobic. So there are octyl or phenyl groups attached to this inert matrix. So because of which this is actually an hydrophobic matrix. So this kind of situation is there. There are hydrophobic ligands attached to the chromatographic support, because of which the proteins where there are hydrophobic zones, these proteins can attach to this matrix, attach to this resin. And how do we elute out the protein? So for elution we use decreasing salt concentration or increasing organic solvent concentration. So by increasing the organic solvent concentration, we can elute out our protein of interest from the column. And the last thing we will discuss today will be dialysis. So dialysis is a process in which there is a semi-permeable membrane. So this bag that you can see here, this is called a dialysis sac or dialysis bag, and this is made up of a semi-permeable membrane. So what we do is we put the protein solution in this bag, and we use these clips to clip off two ends of this protein bag or this dialysis bag, and then we keep this dialysis bag in a dilute solution. So this is at the start of the dialysis. So what happens that the molecules which are smaller than the particular pore size or than the cutoff of this dialysis bag, the molecules which are smaller, they can come out of this semi-permeable membrane by osmosis, right? So there is again a cutoff of these dialysis sacs, dialysis bag. There is a molecular weight cutoff, or it is called MWCO. So suppose the molecular weight cutoff is 10 kilodalton. So the proteins which are lesser than 10 kilodalton, or any molecule which is lesser than 10 kilodalton, will come out in the solution from the semi-permeable membrane, right? So this is how at the beginning of the dialysis there is concentrated solution inside this dialysis bag, and then after a certain point of time, then what happens, after some long time, then this solution becomes in equilibrium. So here what you have inside the bag, that is your small low molecular weight compounds or the low molecular weight proteins have now come outside this dialysis bag, and this dialysis process is usually used for removal of low molecular weight compounds and also for buffer exchange. So often we have to exchange the buffer of our solution, because suppose we have purified our protein using nickel NTA chromatography. So we have eluted our protein with a particular imidazole concentration. Suppose it is 150 millimolar imidazole concentration where our protein is eluted out. So at 150 millimolar imidazole concentration, so our protein sample now that we have collected from the column, that will have this 150 millimolar imidazole concentration. Now if we need to use the protein or if we need to store the protein, then we need to change this buffer, or we need to remove this imidazole from the protein. So what we do is we put this protein in this kind of a dialysis bag, put this protein solution sample in this dialysis bag, and then we allow it to dialyze so for a long time, for example for overnight, in a buffer, and the external buffer that we put here does not have any imidazole. So then what will happen, after a certain point of time, this imidazole molecules, since they will be lesser than 10 kilodalton or lesser than the cutoff of this dialysis bag, they will come out in the solution, and then it will be in an equilibrium. So our protein, our buffer of this protein, the buffer of our protein can be exchanged in this way. Now I have discussed these chromatographic techniques, but after these chromatographic techniques, how do we check how much amount of protein is being eluted out? How do we check the quality of the protein? So for that, the experiments that we do is UV absorption and Bradford assay. So Bradford assay is a Kumasi blue G250 dye, which is used to spectrophotometrically detect what is the concentration of the protein, what is the quality of the protein, and UV absorption at 280 nanometer, right? At 280 nanometer, the UV absorption can also be monitored to detect the concentration of the protein which is eluted or separated after chromatographic techniques. So that's all about today's discussion. Now let's move to our question answers. First question is, a particular protein can be separated from other contaminating proteins by so you can understand that this correct answer will be chromatographic techniques. Next question. During gel filtration chromatography, separation occurs on the basis of, I already discussed gel filtration chromatography. What is the basis of separation in gel filtration chromatography? Charge, pi, shape and charge of the protein, or shape and molecular weight of the protein? So the correct answer is shape and molecular weight of the protein. Now next question. Matrix used for gel filtration varies according to their. So for gel filtration chromatography, the proteins will be separated according to their molecular weight. And how they are separated according to the molecular weight? By the pore size of the column. So the pore size of the column will determine which proteins will go into the pores or which proteins will not go into the pores and come out first. So the correct answer is pore size. So the matrix has to be selected according to the pore size. Next question, in case of thin layer chromatography, what is the stationary phase? For the stationary phase is usually silica for thin layer chromatography. Correct answer is silica. Next question, void volume in a gel chromatography column refers to space within the matrix pore, volume of the column which is unoccupied by the matrix, and volume of the protein added to the column. So the correct answer here, the void volume, is the volume of the column which is unoccupied by the matrix granules. That is V0 or the void volume. Next question. So there are three proteins here, protein A, B and C, which have these molecular weights 13.3, 27.5 and 63.4. So you can see that there is a difference in the molecular weights, and the pi of the proteins are 8.7 and 9.5. So the pi of these proteins are very close to each other. So therefore we cannot use a separation method which relies on the pi or the pH, that means on ion exchange chromatography. So we need to rely on the size. So size based separation or chromatography has to be done. So therefore the appropriate method of separation for these proteins will be gel filtration chromatography. So that is the correct answer, because these proteins differ hugely in their molecular weight or size. Next question, affinity chromatography deals with specific binding of a protein. protein DNA interaction, protein carbohydrate interaction, or charge of the protein. Now, as I mentioned, affinity chromatography is the process where the specific affinity of the protein with another molecule is taken into consideration. So the correct answer is A, specific binding of a protein with another molecule. Next question. Once a protein comes out of the gel filtration column, how is it monitored? What are the techniques used to monitor the protein? Is it pH paper? Is it pH meter, UV spectrophotometer, or viscometer? So the correct answer is UV spectrophotometer to measure the absorbance of the protein. So once it is coming out of the gel filtration column, then the protein is measured, the UV absorbance of the protein is measured by a UV spectrophotometer which is attached with the instrument. Next question. Carboxymethyl matrix and DEAE, diethyl aminoethyl matrix, they are generally considered as which of the following? So we need to now check the charges on this matrix. So carboxymethyl, carboxy means it's a negatively charged group, right? So carboxymethyl matrix is a negatively charged matrix. And DEAE, which is diethyl aminoethyl, so amino group is there, which makes the matrix positively charged matrix. Now if it is a negatively charged matrix, then it is a cation exchanger, and if it is a positively charged matrix, then it is an anion exchanger. So in a negatively charged matrix, cations will bind, and by increasing the salt concentration of the buffer, so by passing 1 molar NaCl, we can elute out the bound protein, right? So by high concentration of salt, we can elute out the protein. So therefore the positively charged protein will bind. So this is a cation exchanger, and negatively charged protein will bind to the DEAE matrix, which makes it a anion exchanger. So the correct answer would be CM as cation exchange and DEAE as anion exchange. So while answering these types of questions, please be very careful. Please always consider what is the charge on this matrix, and then what is the ion that it can exchange. If it is a negatively charged matrix, it can exchange positively charged ions, cations, or if it is a negatively positively charged matrix, it can exchange negatively charged proteins or molecules which are anions. Next question. Before loading the protein onto the chromatography column, which are the following treatments that should be done? So before loading the protein, we should always equilibrate the column with buffer, which will be used as a mobile phase. So this one is the correct answer. There should be equilibration of the column. Next question. So now here are 4 proteins, A, B, C and X. The molecular weight and the PI of these proteins are known. Now how do we isolate X? the molecular weight of A, B, C and D are given. F, C and X are given. Molecular weight of A is 15.1, B 25.7, C 24.2 and X is 23.8. So the molecular weights are very similar for all of these 4 proteins. Now look at the PI. The PI is 3.0, 8.7, 7.3 and 4.3. So these 2 proteins, that is B and C, they have very close PI. They have also very close molecular weights. Now how do we isolate our protein X? So X has a PI of 4.3. So therefore we can use the ion exchange, because at a particular pH it will be, suppose if we use a pH of 7, then this pH will be greater than the PI, right? So therefore the protein will be negatively charged. So we can use ion exchange. Now we have to use a second method as well here. So what we do is we use ion exchange followed by gel filtration chromatography. So we use 2 chromatographic methods because we need to separate the proteins based on their molecular weight as well as their PI. So that will ensure a better separation. So therefore we will use ion exchange followed by gel filtration chromatography. So protein X has a molecular weight of 23.8 kilodalton. So we can use gel filtration chromatography, which will separate protein X from protein A, because at pH 7, for example, if we carry out the ion exchange, then at pH 7 protein A will also be positively charged. pH is greater than PI, right? So therefore if pH is greater than PI, protein will be negatively charged. So at pH 7 A will be negatively charged, X will also be negatively charged. So these 2 proteins will come out together, and we will not be able to isolate these 2 proteins based on the ion exchange chromatography. Now, but B and C, these 2 proteins, they will be positively charged. They will be positively charged at pH 7, so they will be coming out in the flow through. Now, after we have isolated A and X together, now we need to isolate A and X again based on their molecular weight. So since A has a lesser molecular weight than X, therefore they will be separated in the size exclusion column. So therefore that is why we need to use 2 chromatographic methods here, ion exchange and gel filtration. Ion exchange followed by gel filtration chromatography. Okay, next question. So if the protein has a 6x histidine tag and it is overexpressed in bacterial culture. Now after purifying by nickel NTA chromatography, we run the eluted fraction in SDS page, but there are additional proteins also with your protein of interest which we can observe in the gel, SDS page gel. So what is the reason for this kind of additional proteins? That means the contaminant proteins. Why are they there? So the possible reason can be that some bacterial proteins are also having repeats of histidine residues. So this is a common thing that can be present. Histidine is a common amino acid. So of course our bacterial protein can also have some proteins which have histidine residues. So these histidine residues will therefore also allow the other proteins to attach to the nickel NTA matrix by this coordination complex formation, right? So because of which, when we elute proteins with increasing concentration of imidazole, with our protein, these other proteins might also come or be eluted out, which also have histidine repeats. So these is possibly due to the bacterial proteins which have the histidine residues. That is the correct answer. Next question. Now this is a question which asks, why is it advisable to degas the mobile phase in case of gel filtration? So degassing means what? So degassing means removing the bubbles from any solution, any buffer that we are using for gel filtration column. So we remove the buffer and we run the buffer. We remove the bubbles from the buffer and then we run the buffer in gel filtration. So why is it recommended? Because under high pressure, air trapped in the mobile phase can be removed, should be removed, so that the flow of the mobile phase is not hindered. And if there are bubbles in the mobile phase, then these bubbles will actually increase the pressure and it will damage the column. So therefore it is always recommended to degas the mobile phase. That is the correct answer. Otherwise it would hinder the flow of the mobile phase through the matrix, and it will also not be good for the column. Next question. In an FPLC system, the flow rate is maintained at 2 to 5 ml per minute. What if we increase the flow rate? So if the flow rate is increased, it will lead to breakage of the column. So the correct answer is C, column breakage. And the last question for today. When not in use, the gel filtration column and the tubes in the FPLC system should be stored in 20% ethanol. So that is a storage solution that we use for our column and all the tubes of the FPLC system, which is good for the column. So it does not allow the column to dry up in 20% ethanol solution. Okay. Sorry. This is the last question for today. Right. So that is the correct answer, 20% ethanol. And that's all for today. With this I end today's session. We will meet again in the next session on next Tuesday from 6 to 8 p.m. If you have any doubts, you can always post the queries. You can let me know the queries in my email ID or in the discussion forum of the course. And thank you for watching.